Muy buenos días a todos. Bueno, hoy tenemos una conferencia más de los seminarios de Frontiers in Genomics. Como ya saben, este semestre y probablemente el que viene lo haremos todo por la misma vía remota, es decir, en línea. Y para nosotros es hoy un gran placer de presentar a la doctora Tuuli Lappalainen, que es profesora de la Universidad de Colombia y también es miembro del New York Genome Center. La doctora Tuuli es originaria de Finlandia y obtuvo su doctorado en la Universidad de Helsinki, después hizo un postdoctorado en la Universidad de Ginebra y luego en Stanford, en la Universidad de Stanford con el doctor Carlos Bustamante también. El trabajo de su grupo se orienta hacia estudiar la variación genética funcional en poblaciones humanas y particularmente en estudiar cómo las variantes genómicas de un mismo gen pueden afectar el transcriptoma y cómo estos cambios luego contribuyen al riesgo de desarrollar algunas enfermedades. Su trabajo es un trabajo esencialmente bioinformático en el cual se analizan grandes bases de datos genómicos y transcriptómicos y se integran justamente toda esa información para entenderla y dar una idea de cómo es que justamente toda esta variación genética puede afectar a la población humana. En el tema de hoy, justamente, pues es lo que les acabo de decir, que es cómo la variación del genoma humano puede llevar a variaciones en el transcriptoma. Tuuli, thank you for joining us today, and really, yesterday was very interesting, and I'm sure that today will be even more interesting.
Thank you very much. Thanks for having me. And it's unfortunate that I can't be there in person, but this is a good second best. So my lab works on functional variation in the human genome, and in particular integration of large genome and transcriptome datasets to understand this. And today I want to tell you about some of the published work that we have been doing in this space recently, and then move on to some recent work looking at the long-read transcriptome. So during the past 10 or 15 years, the human genetics community has been super successful in mapping genetic variants that associate to different common and complex and rare diseases and traits in humans. But despite this very rich catalog of huge numbers of associations, the challenge is that these associations alone don't tell us very much about the biological mechanisms that mediate these associations. And one of the ways to try to bridge this gap is to analyze how genetic variants associate to gene expression level and other traits of the transcriptome or other molecular traits of the cell to basically build this sort of a trajectory from teeny tiny genetic perturbations to highly complex human traits. And this type of integration of genome and transcriptome sequencing data allows a highly scalable interpretation of GWAS loci, and is particularly important in this space because of the fact that about 90% of GWAS associations are in the non-coding space of the genome. That was initially, when GWAS started to be done in a large scale, it was a bit of an unpleasant surprise when people noticed that, like, oh, this is going to be very hard, that is not going to be obvious biological lessons just falling out of these data, that we really need to do a lot of work to understand the regulatory effects of genetic variants. In addition to that, these kinds of data of genetic associations to molecular traits provides a lot of core information about the regulatory code of the genome, how these naturally existing variants in the population affect gene regulation, gives us information that is complementary to those that we can get from experimental perturbations of the genome. Additionally, there is a lot of data in these kinds of data sets about the functional genetic architecture of human traits, and thinking about sort of what kinds of variants and what kinds of functional mechanisms actually contribute to genetic associations for human traits. For example, I'm particularly interested in analyzing gene dosage as the sort of like a unifying point of convergence of both common and rare variants affecting human traits, and also genetic and environmental effects of underlying human traits. I'm not going to talk about that so much today. It was a bit of a topic for another day. And the same applies to the analysis of the personal genome with transcriptome data. So now in the clinical genetic space, it's becoming more and more common to use genome sequencing or exome sequencing to diagnose patients and actually understand what is the variant and the gene that is underlying a patient's severe disease. But since it's so difficult to sometimes interpret the genome and distinguish which of these variants are actually the functional ones that severely disrupt gene function, the transcriptome data can help in that. And us and others have been analyzing or developing methods to achieve this. So this general field of analyzing functional effects of genetic variants is really actually at the intersection of traditionally surprisingly distinct areas of genetics and genomics. So there is, of course, the sort of many fields that focus on genetic variation in human populations, quantitative genetics, population genetics, thinking about the origins and the consequences of genetic variation. But then on the molecular genomics side, projects like ENCODE and others have been doing a lot of work just understanding the sort of function of the genome. How does the blueprint of the genome produce a functional cell and a functional organism, but not necessarily thinking so much about genetic variants. So we kind of borrow a lot of the statistical methods from like the GWAS field and a lot of the molecular assays and interest in function from molecular genomics and put these two things together. So when it comes to sort of a scalable approaches to study molecular effects of genetic variants, from a computational or sort of like a big data perspective, there's kind of four things that we can do. So the first two would be, the first would be sort of like something that we probably have all done, that you have some genetic variant of interest and you go to your favorite genome browser and you kind of like see where is it, what are the genes nearby, does this overlap any enhancers, is it a conserved position? But this is a very sort of ad hoc, imprecise approach. There are also more advanced predictive models to try to use these types of information, for example, from annotations and conservations to produce different types of scores for variant functionality. And this is, of course, where we want to go. We would want to be able to predict genome function just from the sort of annotations and the genome sequence alone. But the problem is that when it comes to regulatory variants, we're really not there yet. We cannot accurately predict variant function. So that's why my lab and many others have been kind of taking the approach of like basically using empirical data of like associating, linking genetic variants to molecular phenotypes in various different ways. So the most classical approach is the kind of QTL mapping approach, where we simply look for genetic associations for molecular phenotypes, such as gene expression, to find those variants that are associated to changes in some regulatory features of the genome. And then more in the rare disease space, which I'm not going to talk about today so much, we can look for sort of rare variants with unusually strong effects on gene expression by kind of detecting outliers and linking those to genetic variants. In addition to these computational approaches, there is, of course, now an expanding, increasingly rich toolkit of experimental methods to perturb genomes and see what happens. This includes, of course, the various different varieties of the CRISPR toolkit, and also MPRA and other massively parallel systems. And we use these methods in the lab as well, quite a bit, actually. With some papers kind of in the published or preprint space and others in the works, and I think that this is integrating these kinds of experimental perturbations and observational studies of genetic variation in human populations is going to be extremely important going forward. But there is still, when it comes to these experimental methods, the sort of scale and interpretability still needs to improve quite a bit. So that's why today I'm going to sort of focus especially on the QTL mapping space and what we have learned on regulatory variation using that approach. So the largest study to use the QTL mapping methods to understand regulatory variance is the genotype tissue expression, or GTEx study. So GTEx was launched about 10 years ago with the aim to build a comprehensive atlas of genetic regulatory variance across a diverse set of human tissues using postmortem donors. And over the years, GTEx has published a bunch of papers from the different phases of the project, and the data has of course been used widely by the rest of the community as well. And I've been part of GTEx analysis group since the very beginning, from like about 2011 when I was still a young postdoc, and now I was fortunate to be co-leading the final set of GTEx papers that we published a couple of months ago in Science. So I'll tell you a little bit about some of the key discoveries of those science papers, and then move on to some unpublished work. So in this the V8 release of GTEx that these papers were based on, we had altogether over 17,000 tissue samples from over 50 tissues of 838 donors. So basically a bunch of multiple different tissues from each of these postmortem donors, and then RNA sequencing data from all the tissue samples, and then genome sequencing data from each of the donors. And this allowed us to do QTL mapping, so looking for genetic associations between genetic variants and gene expression and splicing, specifically focusing on the cis regulatory space, so looking for each gene, looking for genetic associations in the 1 megabase region surrounding that gene. So here what the middle plot, the scatter plot, shows is just the numbers of associations that we find. So GTEx has not every tissue is collected from every sample due to quality and consent and other things. So that's why we have the tissues that are shown in different colors vary a little bit in their sample size. But so we can see that there is, of course, there is a correlation between the discoveries and sample size just because of power, but especially in the largest tissues, we really find like well over 10,000 genes that have at least one eQTL and close to about 5,000 genes or so that have a splicing QTL. So a really, really large number of these regulatory associations, reinforcing the message that we have known from previous studies, that these kinds of genetic associations to molecular traits of the transcriptome are really, really widespread in the genome. They are everywhere. We can use this rich catalog to then interrogate what kinds of functional mechanisms may be driving some of these regulatory associations by looking at the enrichment of these QTL association signals in different annotations of the genome. And one thing that is very obvious in this data is that splicing QTLs are highly enriched in the transcript regions of the genome, which is hardly surprising. It's a post-transcriptional regulatory mechanism primarily. Also, eQTLs have high enrichments in sort of post-transcriptional elements, but then also in classical transcriptional regulatory elements such as enhancers and promoters. Although one thing that is kind of easy to see in these data is that those enrichments are relatively modest, and we believe that this is just simply due to the fact that enhancers and promoters, these annotations are relatively large elements of the genome where only a tiny subset of the variants that are annotated as being in an enhancer or in a promoter or promoter flanking regions, only a tiny proportion of those variants are actually functional. So given the sort of like our inability to do nucleotide level annotation of regulatory potential of these variants kind of dilutes the signal compared to something like, let's say, variants in canonical splice sites or stop gain variants, where it's really kind of nucleotide precision sort of estimation of this variant being likely functional. GTEx being a multi-tissue dataset, of course, makes it possible for us and a major point of interest to analyze how these regulatory variants may vary between different tissues. Of course, the genome is the same in all of our cells and tissues, unless you have cancer. But the function of the genome and the activity of different regulatory variants is not necessarily the same. But actually, what we see in the data is that actually the extent of sharing of cis-QTLs is actually quite high for a lot of the tissues. So there is some structure in the data that is easy to see. Different brain sub tissues or subregions cluster together, as do a large number of sort of internal organs, going from like stomach to lung to heart, etc. And this kind of like high degree of sharing is not exactly something that we expected or knew about when GTEx was launched. It is notable, though, that there are outliers. For example, whole blood, that is unsurprisingly, just due to practical reasons, one of the most common tissues to be used in different types of functional genomics analysis, is an outlier and not a super good proxy tissue for many other tissues and organs. Again, if we go and look at enrichments of these different types of tissue shared and tissue specific variants in different functional elements, we can start to understand what are the functional mechanisms that make certain variants to have more tissue shared versus more tissue specific effects. And we see both for eQTLs and splicing QTLs that basically elements that are likely to have post-transcriptional effects. So they're in transcribed regions are enriched for tissue shared effects, which makes certain sense, but not necessarily, again, something that we knew so clearly before. And this enrichment probably contributes to the fact that splicing QTLs in general are more shared between tissues than eQTLs, that they are enriched for post-transcriptional processes, and post-transcriptional processes are enriched for tissue shared mechanisms. And then for some of the kind of transcriptional regulatory elements, open chromatin, transcription factor binding motif, we see an enrichment for tissue specific effects. But one of the important factors to understand and consider when one thinks about this extensive catalog of regulatory variants and gene expression across different tissues is that, of course, these tissues are composed of multiple different cell types. But when GTEx was started over 10 years ago, there were no single cell technologies around, and even now, despite Human Cell Atlas, we are very far from being able to do something like GTEx actually at a single cell level, or analyzing all of these things at the level of specific cell types. But to be able to get insights into the cell type composition in these samples, now that we understand even better than we did many years ago how important cell type composition is, we did an in silico analysis of enrichment of 64 cell types in each of the GTEx samples based on gene expression signatures. So this is not exactly cell type deconvolution, but just estimation of kind of how highly these different 64 reference cell types are in different tissues. And this just shows the kind of median enrichment in different tissues for these cell types. And in general, we see patterns that we would expect. We see enrichment of myocytes in muscle, hepatocytes in liver, keratinocytes in skin, etc. But an interesting thing emerges from this data when we look at the sharing of, so it's just pairwise similarity of different tissues based on this matrix of cell types. So this is in many ways a very, very similar plot that what I had on the previous slide for cis-QTL sharing, looking at just effects of genetic variants. And here we're looking at cell type composition. And actually these two matrices are 90% correlated. And what this means is that tissue sharing of eQTLs is actually likely largely shared by different tissues sharing the same cell types. So that, for example, if you have an eQTL that is specific to epithelial cells, then that eQTL is going going to show up in all the tissues that have a high proportion of epithelial cells. And in this way, this really highlights the fact how even us geneticists who don't necessarily think about molecular biology of the cell so much, but if we want to understand tissue-specific activity of different genetic variants, we really need to pay close attention to the cell type composition, and much more work is needed in this space going forward to be able to fully characterize this with, of course, emerging single cell technologies. So in addition to looking at that kind of like just sort of median enrichment of different cell types in different GTEx tissues, we can also look at the variation of those enrichments for the individuals in a given tissue. So such variation in cell type composition, which can be partially biological, but probably also technical, depending on how the tissue sort of specimens are collected, that could sometimes be considered like a technical artifact or a nuisance. But in our analysis of genetic variants, we can actually use that to our benefit to map cell type interacting QTLs. So in these analyses, if we look at the first scatter plot as an example, so this is skin samples with an eQTL for this between this particular variant and a given gene. But what we can see is that the effect size of that eQTL is stronger in individuals that have a higher enrichment of keratinocytes in their skin. What this means is that this regulatory variant is probably active in keratinocytes or in another cell type that is positively correlated to keratinocytes. And we wouldn't call this eQTL a keratinocyte specific eQTL, because like the cell type composition of in a given tissue, these like different cell types are highly correlated to each other. So we can't know for sure that this is actually driven by keratinocytes and not something else that is correlated to that. But still, what this data tells us is that this QTL has variable effect sizes in different cell types and is not equally active in all the cell types in the skin. And we can do this for cis-QTLs and cis-splicing QTLs. And I actually have old paper references here. I just noticed. So these are both published a couple of months ago in Science, including a companion paper focusing on these cell type interacting QTLs, led by a former postdoc in my lab. But so anyway, so we did this analysis for seven different cell types across multiple tissues where these cell types are present, and identified up to over 1,000 cell type interacting QTLs. So when it comes to using these data for better interpret functional mechanisms of GWAS loci for different complex traits and diseases, the most classical approach is to basically look at what we call co-localization. So when you have a given locus with a GWAS association, you look to see if there is an eQTL or an sQTL in the same locus. And then you analyze that using sort of different statistical approaches to see if those two associations seem to be driven by the same causal variant. Like if the association landscapes really seem to kind of like mirror each other. And if they share the same causal variant, that puts forward a hypothesis, still not proof, but a hypothesis that that this regulatory association, this molecular change in expression or splicing, could be driving that disease association and causally contributing to that. So doing this analysis in GTEx, we found thousands of loci where we find indication of co-localization, and thus are able to build a lot of potential hypotheses, of course, on mechanisms of different diseases. But an interesting thing emerges when we start to look into our cell type interacting QTLs, where it's not uncommon that when we just do the sort of like vanilla tissue level eQTL co-localization, that we have situations like this, where we have a nice platelet count, GWAS association, and then there is an eQTL in this locus in whole blood, but it doesn't actually co-localize. These association signals don't look very much alike, and it's the sort of our analysis approaches tell us that they don't seem to share the same causal variant. Kind of like, yeah, move on. But when we look at our neutrophil cell type interacting eQTLs, we actually do find an association landscape that closely resembles the platelet count GWAS association with a high posterior probability for a shared causal variant and colocalization. And what is going on in this locus and in many other loci is that when we analyze a tissue level eQTL data, we actually have multiple independent signals derived from different cell types that are kind of smashed together into one blob of a signal that kind of like lacks the resolution to be really easy to interpret and easy to analyze in the context of GWAS associations. And with this cell type interaction approach, we're able to sort of like pull apart those contributions from different cell types and better interpret these regulatory associations. And we can actually see that we find a substantial number of additional co-localizing loci when we look at our cell type interacting QTLs. So another interesting question that we're now able to answer is concerns the sort of convergence of different genetic associations for complex traits. So what I mean by this is that at this point in GTEx data and in general in sort of human genetic data sets around, we often have a situation where we actually have identified multiple genetic variants affecting the function of a given gene. So in GTEx, we often have not just a single eQTL for a given gene, but multiple independent signals that are not in LD with each other. And often we have also an eQTL and an cis-QTL affecting the same gene, just sort of like a different transcriptome trait, but kind of, yeah, independent effects on a given gene. And then additionally, there are other data sets, such as from the UK Biobank and elsewhere, where we have rare coding variant associations for complex traits. And this gives rise to the question that, like, if one of these variants is associated to some GWAS trait, are the others as well? And if that is the case, if there is this kind of like concordance between functional variants for the same gene, this would suggest that that these are really kind of like mediated by functional effects on this particular gene, and that kind of like different types of functional hits on that gene really kind of mediate the disease association. So, so we did this analysis in multiple different ways. To make a long story short, looking at when we have multiple cis-EQTLs per gene, if we have cis-EQTLs and splicing QTLs for the same gene, or if we have a rare coding variant and a common regulatory variant affecting the same gene, we see a significant strong signal of this kind of convergence of GWAS effects at the gene level, which is really highlighting the fact that there is indeed this kind of that sort of gene dosage and functional effects of the gene is sort of like a key functional point where that can help us to better interpret individual associations that we may have around these genes and around the genome. So to kind of summarize this part where we are and what happens next after GTEx, that is now basically over, we've really built a comprehensive map of the extremely common regulatory variation that we have in human populations. The cis-EQTL and splicing QTL catalog across human tissues in Europeans is essentially complete. It's not that we have every single tissue, but we have a lot of them. And this kind of takes away a lot of the need from the community to kind of, if you want to analyze regulatory variants in liver, you just check GTEx. If you want to analyze skin, check GTEx. And thus empowering a lot of future research. But there are still a lot of gaps that we need to kind of fulfill as a community. So I've highlighted several times now how important it is to get to specific cell types from that tissue level and do eQTL mapping in a cell type specific manner. So GTEx focus on cis-EQTLs and cis-QTLs, so basically those genetic variants in the cis-regulatory space around these genes. We also did trans-QTL mapping, but it's still challenging. It requires a lot more samples, like several thousands, and potentially also single cell resolution to deal with some of the biases. So this is something that the community still needs to push forward, because these kinds of trans associations between genetic variants and genes completely elsewhere in the chromosome can be very informative concerning sort of pathway and regulatory network level effects that genetic variants can have. Here we focus on expression and splicing that we can quite easily quantify from transcriptome data. And transcriptome RNA sequencing is of course the most scalable functional genomics approach, but other molecular phenotypes are very important as well. And there has been very exciting work that has been done, for example, looking at attack-seq data and this kind of chromatin QTLs, and those studies should be expanded. GTEx is a European-centric or European ancestry-centric data set. So it's an American sample. So it's about 85% European ancestry and then a mix of others. And now that GWAS studies are increasingly and very necessarily so becoming more diverse and including more non-European samples, it's essential that these functional genomics data sets to interpret those GWAS are also kind of like catch up and provide the data to be able to interpret those associations. So this is also something that us and others are pushing forward. And then importantly, this kind of a QTL mapping approach, it is a population association approach, and it's a very useful and a scalable approach to understand regulatory variants in the genome. But as I mentioned in the beginning, now we have an increasingly rich experimental toolkit to basically probe at these questions with a completely different set of approaches, and integrating these things is going to be very important going forward. And all of these questions are something that we work on in the lab. But next I want to tell you about a study that kind of barks at the tree of these other molecular phenotypes, and not actually still focusing on the transcriptomes, but thinking about better transcriptomes. Because this short read RNA sequencing data from the Illumina platform, it's been great. It's served us very well for 10 years, enabled a lot of research, a lot of insights. But these short reads have limitations. And it's very important to understand that actually, when we think about the transcriptome, the real biological units and the functional molecules of the transcriptome are transcripts. It's not genes. And even though we know this, still, for example, eQTL mapping and many analyses really focus on gene expression levels instead of transcripts. And this is partially due to the fact that transcript detection and quantification from Illumina RNA sequencing data is still, it's somewhat challenging. The human gene and transcript annotation is still far from perfect, even though it's the most comprehensive one that we have of mammals. And then to quantify these transcripts from short read data is quite imperfect still. We also have typically a relatively poor resolution into different types of transcript changes. So for example, the splicing QTL mapping that we have done in GTEx is kind of like the quantitative trait there is this kind of like some kind of a change in splicing. It's sort of like a detection of sort of intron-exon junctions. And it's not this kind of like a, let's say, 21 base pair extension of the 3' end of the 7th exon. We don't get that kind of resolution into what actually changes in the transcripts and what actually then matters potentially for, let's say, GWAS associations. And another limitation is inability to analyze allele-specific transcript structure. And I'll tell you in a couple of minutes what that means. But basically, these caveats and the expanding sort of capabilities of doing long-read RNA sequencing empowered us to study, create a large number of long-read sequencing data from a bunch of GTEx samples. And the kind of key sort of limitations that have been hindering large-scale long-read RNA sequencing data thus far, and that are now sort of manageable, or at least something that we didn't find as major limitations, is many of the long-read technologies need a huge amount of input material. But GTEx is not just a data set, it's also a biobank. One can request access to these samples, including cell lines, where you can get very high quality RNA in relatively large amounts, and also those the needs for input material in terms of quantity have gone down. Then cost has, of course, been prohibitive for to be able to get enough reads to be able to quantify transcripts. It's not enough for us to just kind of like have one transcript and do this kind of like qualitative annotation of different transcripts. We want to be able to quantify them, and for that you really need reads. And this is still quite challenging for PacBio, but Oxford Nanopore Technologies, their kind of yield and throughput has increased quite significantly, and for a pretty reasonable cost, you can actually get similar levels of coverage as you could get for Illumina. And then sequencing errors are an issue, especially for ONT. But this is not necessarily a huge issue in transcriptome analysis if you kind of know what you're doing and analyze your data appropriately. We're not trying to do SNP genotyping here just to analyze some of the transcriptome. It actually works almost surprisingly well. So we basically formed a collaboration with a bunch of folks. So all of this study and this work was led by Daphne Klinos, who's a postdoc in my lab. And we collaborated with Dan MacArthur and Beryl Cummings, who both used to be at the Broad and have since moved on. And then also collaborated with Oxford Nanopore themselves. Their US office and lab is on the same floor with us. They're leasing space from the New York Genome Center. So that has made this collaboration very, very easy and smooth and pleasant. So the data that we analyzed here was consisted of 70 samples with RNA sequencing data using mainly the cDNA PCR protocol from Oxford Nanopore. So you may know that ONT platform also allows direct RNA sequencing, which is quite exciting and unique. But the direct RNA-seq yield is so low that it's not very feasible. It's super difficult to do. So cDNA sequencing works much better. And since these are GTEx samples, we have phase whole genome sequencing data and Illumina RNA sequencing data from these samples as well. And altogether, these 70 samples cover 43 donors and 15 different tissues, with like a handful of different tissues, different donors per tissue. There is also some additional data sets that I'm not going to talk about today. And then we analyze these data with a relatively sort of basic standard ONT transcriptome analysis pipeline. Luckily, these tools are now starting to be in place. But if there are any tool developers in the audience, this is a very, very kind of like fertile ground where there is a lot of need for improved tool development, both in terms of accuracy of the tools and the scalability of the tools. This is a very large data set, and just getting some of these tools to run was a challenge. They've been sort of designed for a few samples, and we had like over 17. So what we got from these samples was a median of 5 million aligned reads per sample. So this is a little bit lower than what you would typically have from Illumina, which would be maybe like 20 million or something. But these are long reads that contain a lot more information than those short reads. The median read length, the aligned read length, was 800 base pairs. So these are not always full transcripts. They may be like fragments of transcripts, but they are still long reads that carry a lot of information about the transcript structure. So one of the first things that we did was to do novel transcript detection from the data using the FLAIR pipeline, discovering over 100,000 novel transcripts. And after we sort of like filtered this data down using several kind of sophisticated criteria to get sort of a high confidence set of 14,000 novel transcripts, an exciting thing that we observed was that actually the tissue clustering of the samples was kind of cleaner and clearer with these novel transcripts compared to annotated transcripts, which really shows that these are not just some sort of like garbage coming out of our pipeline. This really carries true biological information. And an exciting opportunity to validate this data was provided by the fact that one of the GTEx companion papers looked into proteome data from GTEx samples using mass spectrometry. So this is from Mike Snyder's lab, and it was a great opportunity to collaborate with them to basically see what are the validation rates, how often do we find the peptides that we would expect to see based on our transcript annotations in the mass spec data. And excitingly, the validation rate of our novel transcripts was almost the same as that for the annotated transcripts, really showing that these novel transcripts are they're high quality. And obviously we will be providing these transcript models when we preprint the data. This is a relatively significant sort of expansion of the annotated space of the genome. But what we actually wanted to focus on in this study was genetic effects on the transcriptome. And here, the analysis of allele-specific transcript structure is really a unique opportunity provided by these long reads. So my lab and many others have been analyzing allele-specific expression for a long time. And the way this analysis works on the left-hand side is that you basically get from your genotype data from whole genome sequencing or genotyping, you identify heterozygous variants in a given sample. You look at one sample at a time. And then you can go to your RNA sequencing data, and here short reads work just fine. And then simply count how often do you observe each of the two alleles, with the null expectation, of course, being a 50-50 ratio. But it's not uncommon that we see that actually there is sort of like more reads coming from one of the two alleles, which is suggesting that one of the haplotypes is expressed at a higher level than the other one. And this is almost always driven by the individual being heterozygous for some cis-regulatory variant somewhere in the cis-regulatory space of that genome that is driving this higher expression from one of the haplotypes. But when it comes to transcript structure and kind of allele-specific effects, haplotype differences in that we really cannot study from short reads, because basically here in the example in the middle, the GC variant that we are like using to separate like what are the reads coming from each of the two haplotypes, they're so short that they can only sort of like capture what is going on right around that variant, and you are unable to see this change in transcript structure that is in the other end of the gene. But with long reads that kind of like cover those heterozygous variants to separate the two haplotypes, and then much of the rest of the gene, they carry that information to be able to do allele-specific transcript structure analysis. And again, the expectation is that these are very largely driven by heterozygous genetic variants when we see these differences, us and others have shown this before. And since this is a largely genetically driven phenomenon, it really complements eQTL and cis-QTL mapping nicely. These are kind of two orthogonal approaches to approach cis-regulatory genetic effects on the transcriptome. And then also there are some interesting possibilities for rare variant analysis. We have previously done work on genomic imprinting and X chromosome inactivation and other things using allele-specific expression, but I'm not going to talk about that today. But so first, to be able to analyze allele-specific transcript structure, we had to develop a tool to do that in a scalable manner for long reads. So we built a tool that we call Laurels that works such that when our transcript quantification method FLAIR first has assigned every read to a transcript, then we use Laurels to assign the annotated reads to the two haplotypes based on an allele at a heterozygous site, accounting for some of the properties of the ONT data. data. And then we have this kind of contingency tables that we can analyze if the distribution of reads to different transcripts is different between the two haplotypes. And then of course we can also do allele-specific expression from the same data, just summing up the reads. And my lab has published a lot of tools for allele-specific expression analysis from short reads before, so this is kind of building on that. So doing this analysis, we find over 1,000 genes with significant allele-specific expression in at least one sample, and 152 for allele-specific transcript structure. This difference is not necessarily biological, or it's difficult to say, because allele-specific transcript structure is a more complex phenomenon, and we kind of need more data to be able to see those significant effects. But first, what we wanted to do was to kind of like validate if these patterns make any sense and are consistent with previous data. So leveraging the idea that so if we take splicing QTLs and eQTLs mapped in GTEx, we should be able to observe that heterozygous individuals, where we kind of expect the two haplotypes to differ either in their transcript structure or in their expression levels, should show an enrichment of these allelic events compared to homozygous. This would basically allow us to kind of like validate our call. And this is indeed what we see if we look at both eQTLs and allele-specific expression, and cis-QTLs and allele-specific transcript structure. We see that heterozygous individuals are enriched for this signal. So they kind of manifest in these regulatory variants manifest in allelic expression and transcript structure, as we would expect. But we can also study these two types of data, not just as distinct things, but jointly, and asking for a given gene in a given sample, if we see allele-specific transcript structure, do we see allele-specific expression, and the other way around? And we do see that there is a large amount of sharing. There are also exceptions, like in this case, where there's just kind of like no allele-specific expression, but the transcript kind of flips which haplotype prefers which. But actually, this large amount of sharing is a little bit perplexing and somewhat a complicated matter, because in GTEx and many previous papers, when we have done splicing QTL and eQTL mapping and analyzed these variants, we have shown that these variants are almost always distinct. So basically, the variants that lead to changes in splicing and changes in expression, they shouldn't be shared. They should be distinct. But here we see a high degree of sharing. So this was weird, and we started kind of like scratching our heads and decided to go back to our eQTL and splicing QTL data from GTEx to see how they also kind of like manifest in sort of eQTLs, how they manifest in allele-specific transcript structure. And here we saw that there is also a significant signal of basically individuals that are heterozygous for eQTLs having allele-specific transcript structure. And this is, we don't expect this to happen, because they should be distinct. But then we realized that actually this kind of analysis of like eQTLs and cis-QTLs being distinct is based on cis-QTLs that have been mapped with algorithms like Leafcutter that really detect mostly changes in intron exon structure and sort of like exon skipping. But in our data of long reads, we can actually detect a much more diverse set of different types of transcript changes, including changes in, for example, 5' and 3' ends of the transcripts in terms of alternative UTRs, alternative first and last exons, etc. So analyzing this, we can actually see that for cis-QTLs, we see the kind of the only significant enrichment is kind of like exon skipping events. So splicing QTLs manifest in that. That's expected. But for eQTLs, we see that they manifest not just in gene expression changes, but also in basically allele-specific transcript changes that relate to 5' changes in the transcripts. Alternative first exons, alternative 5' UTRs, and such. And this is interesting in the sense that, I mean, 5' end, that's of course where the promoter is. So it kind of like makes sense that some of the changes there would also manifest in expression changes. But basically what this data tells us is that genetic variants that affect expression and splicing are indeed largely independent, but there is this kind of like a joint effects of changes in 5' ends of transcripts and gene expression. So altogether, what we have here is the largest human long-read cDNA sequencing dataset to date, at least as far as I know, and this will be part of the GTEx V9 release this fall or winter. And we have detected tens of thousands of high-quality novel transcripts. We have a new computational pipeline, Laurels, to detect allele-specific events in long-read data. This is tuned for ONT, but should be applicable to PacBio as well. And then with the analysis of these QTLs and cis-QTLs and different types of transcript events shows that how these long reads can help us to resolve the relationship between different types of transcript events and genetic effects on those. And there is also interesting potential for a better rare variant interpretation that I didn't have time to talk about today. So with that, I'd like to acknowledge a bunch of people on behalf of the first part of the talk, the GTEx work, especially François Aguet from the Crowd, who was the lead analyst of this work. This is our GTEx long read project acknowledgement, especially Daphne, Beryl, Garrett, and Daniel, and also folks from ONT. And then this is my lab. Many thanks to them, as well as lab alumni, consortia, and their members, and some of our key collaborators and sources of funding. And thank you for listening, and I'll be happy to take questions. In the meanwhile, I'll just leave this up here. So as a teeny tiny plug, the International Common Disease Alliance is a new endeavor launched by or spearheaded by Eric Lander and Cecilia Lindgren to really advance common disease genetics and build genetic maps, understand the mechanisms of these associations, and develop medical applications. And really sort of like build the roadmap and build the collaborative projects and sort of efforts that are needed to really make advances in this space. So ICDA is now kind of like taking shape. There is a big plenary, 2-day sort of like symposium next week. So if anyone is interested, check out the website and sign up. These working groups are now taking space. And one of the key kind of components in ICDA is not just to kind of like do the science, but also build the joint infrastructure, training, and global equity sort of standards and approaches to really drive forward progress. So I'll stop there and take questions.
Thank you so much. That's a huge effort to see all that effort in terms of what you do and how to coordinate all these people working together.
Yeah, indeed.
So I don't know if we have questions. Let me ask Alfredo. Alfredo, do we have questions? Alfredo is very silent today, so. Oh yeah, there is one here. Let me see. So that's from Alejandra Cervera. Hi, Tuuli. Thanks for a great talk. Do you think that gene expression analysis will shift to long read sequencing and hybrid approach of short and long, or just improved annotations will aid short reads?
So yeah, that's a good question. So, I mean, it's difficult to say at this point, like if the sort of scale and cost of and ease of use of ONT will really come down to the levels where we are with Illumina. I do think that there are pretty many applications where it's kind of like when it comes to certain transcript quantification type of things or a specific transcript structure, is that like it doesn't, like you can try to improve the methods to analyze short reads, but it just doesn't cut it. Like you just don't have that information in the data. So then you cannot really somehow squeeze something that is not there out of it. So I do think that there is a lot of potential. And now also there are increasingly good protocols to start to do ONT at a single cell level. So yeah, I do think that we might be getting close to like a revolution that could be analogous to when we switch from gene expression arrays to RNA-seq, but time will tell.
Okay. So do we have another question or not? Questions are slow to come today.
Yeah.
Not as yesterday, right?
The students asked so many fantastic questions yesterday.
Yesterday we had a lot of questions. I think they don't have any more questions to do today.
Yeah, that's fine.
They got exhausted.
No, I'm good. But,
no, I think it's really interesting all that what you are doing. I mean, it's definitely not what I do, but one can anticipate that it really will bring very new knowledge and a lot of new solutions for problems. There's one question, Kevin Galindo, how feasible do you see somatic cells CRISPR-Cas9 editing of QTLs? How far are we from understanding all the implications of doing it?
Yeah, so I mean, we have done some editing of eQTLs in cell lines, and it works. Of course, the scalability of CRISPR is still, especially if you want to do single base pair edits, is still quite low. So it's not very easy. And of course, the challenge with eQTLs is that, so some of them kind of have pretty big effects on gene expression, like so about of GTEx eQTLs, about 20% has more than twofold effects on gene expression, but it's kind of like for many of these regulatory variants, including some of the disease relevant variants, the effect sizes are so small that it's kind of like, even if you're able to do the edit, how sensitive is your readout to be able to see that difference? And you may actually need to have a large number of replicates. So when we started to do some of the CRISPR work, kind of whatever, 6 years ago or something, I had kind of the perhaps naive thinking that it's like, oh, if we have like the isogenic cell line and then we just edit like a single SNP, we kind of like have the perfect control, and the kind of like the noise will be less than what we have in human populations, because there is, of course, each individual has their own sort of environmental and genetic variation affecting things. But I think that's probably not true. Like, you actually need a lot of replicates in CRISPR analyses. Different isogenic cell lines have like weird differences between them. So kind of like it's going to be challenging, I think.
Certainly.
And I think that maybe the sort of like single SNP editing and creating isogenic cell lines, it's not probably going to be a very scalable approach for analyzing regulatory variants. One needs to kind of like have more sort of multiplex approaches.
Certainly, it looks like that, yes. There's one more question from Ulises Eatl Juarez. He says, great talk. What happened with the transcripts of microRNAs?
So GTEx is a poly-A transcriptome study, and microRNAs are not poly-A, so we basically don't capture those. We have in some previous studies, including the GWAS study back when I was a postdoc, it's now Lappalainen in 2014 Nature. We did basically separate, so like a small RNA sequencing and then poly-A RNA sequencing, and there, one can analyze microRNAs and find some eQTLs for those and stuff. I've always had a little bit of a struggle of like somehow squeezing in interesting things out of those data, but it's probably just me and other people could probably find more things.
So there's one question from Itzel. Could genetic variants generate different responses to processes such as viral infections?
Yes, so we have actually studied this specifically, and this kind of analysis of kind of immune response eQTLs is quite common. There's a number of papers where people have collected primary blood samples and then extracted primary cells, monocytes or T cells or something, and then in vitro expose those to different sort of ligands that mimic viral or bacterial or other infections. And then you can basically see how some genetic variants, like some genetic variants that kind of like were not there under baseline, then they kind of like appear, and also see for some of them that there seems to be really like these are basically gene environment effects, genetic effects on gene expression that occur only under a certain immune stimulated stage of the cells, and then potentially also could affect how the cells respond to the infection. So we have a preprint out, Pran et al., where we actually describe one locus that is an autoimmune disease, Crohn's disease risk variant. that is basically affects the transcriptional network of IRF1 transcription factor specifically under actually bacterial stimulus in this case. And our hypothesis there is that this variant leads to or causes disease risk by kind of dysregulating not the sort of like steady state of the cells, but leads to kind of like an abnormal response to bacterial infection. So yeah, there's a lot of interesting work in that space, and I think that much more should be done, because this is some of the core mechanisms of human disease, I think, can come from those.
Right, totally agree. I can understand better. I'm an immunologist.
I'm not an immunologist, and somehow I've ended up working on these immune projects, and it's kind of like it's hard.
So is there any, from Diego Arturo, says, with this shift towards long reads, where are we in the M2, M2M challenge? Would we start describing mechanisms in a more efficient way?
Yeah, I think so. So a lot of the current tools when it comes to the sort of understanding mechanisms of complex traits, etc., they are very focused on sort of gene expression, like when it comes to single cell RNA sequencing or some of the sort of, let's say, CRISPR-QTL approaches or the MPRAs. But we know from GTEx and other studies that genetic variants that impact splicing have an almost equal enrichment of GWAS associations as eQTLs. So we really cannot kind of disregard or forget regulation of splicing and transcript structure as a mechanism for common disease. And I think that some of the long read approaches are going to be essential for being able to, for example, get proper single cell readouts of like what is actually going on there. And I think that this is like, it's not going to be sort of like, there's no silver bullet in this. Long reads are not going to solve everything, but I think it's a very important aspect that we really need to keep in mind. And it's good that the technology is there now to allow us to do better.
And then from Elsa Peters, are there any known functional eQTLs derived from host microbiome interactions?
Good question. I am not really a microbiome person, but I could imagine that someone has done this. I think especially in the gut, sort of like the kind of Crohn's disease, inflammatory bowel disease people, I think there has been some studies trying to sort of capture eQTLs that change depending on your microbiome, but I don't know.
And from Victor Nieto, great talk. Could it be relevant integrating structural variations into eQTL studies?
Yeah, definitely. So there is, we kind of wanted to do it for these sort of GTEx papers, but we just didn't have, like, our structural variant experts were busy with other stuff, and structural variant calls are part of the GTEx release. So it's there for anyone to study. And from the GTEx mid-phase, so papers published in 2017, there is a paper specifically focusing on those structural variant effects on gene expression. So I think it's important. It's just kind of like, it's a little bit more tricky to analyze because structural variants are more difficult to call. So that's why it's been not so heavily emphasized, but definitely an important aspect.
And from Ellie Sulbaran, she says, is there evidence of genetic variants that have specific effects during development and could give rise to diseases in adulthood? If there is, how often does this happen?
Yeah, so
relevant for studies.
Yeah, so the data of regulatory variants that are specific to different developmental stages is relatively sparse. It's understandably practically difficult to collect enough of fetal samples to be able to do something like a fetal GTEx. However, there is some interesting work that is now starting to emerge from iPSCs that like collected from a large number of individuals and then in vitro differentiated to different, for example, neurals and neuroprogenitor cells. And then forward from that, there's a nice paper in bioRxiv by Daniel Gaffney and Olli Stegel's labs. So they have been looking at this and doing eQTL mapping in neural cells during differentiation, and they do find they find eQTLs, and then they find eQTLs that are not discovered in GTEx, a relatively significant number of those, and also some kind of co-localizations with diseases that have not been observed before. So I think it's still kind of, the data is sparse, but there is quite a lot of interest in this, and I think it will be important, because it's quite well known that a lot of diseases, especially in the psychiatric neurodevelopmental space, it's really kind of developmental stages that are key.
And I think this seems to be the last one.
Okay, well, thank you very much.
Thank you so much for being with us. And I hear that this is your second talk, the 2 of 3 talks. So.
Yeah.
So, well, have some rest before you go to the next one.
Yeah, exactly. No, this was fun, and thanks again for having me. Really enjoyed meeting you and the students, especially yesterday. And yeah.
Well, I'm sure that the students will maybe contact you soon. I mean, they have their next semester and next year is a lab. Is a lab year, so very often actually they reach out to see where they can go. So I'm quite sure that you may get.
Hopefully by that time we're all vaccinated and actually able to go to places.
Exactly. So, well, thank you so much, and I'm very happy that we finally made it.
Yeah, exactly. Thanks a lot.
Okay, thank you so much. Bye-bye.